All right, so let's go ahead and I've been getting some seriously interesting questions. So let's go ahead and go through this one step at a time. We said make sure you understand the kind of the basics that we had talked about in the past, but that this does a great job at summarizing. And let's go ahead and read this. It says, have you ever experienced an odd scratchy feeling or burning sensation on your tongue after eating a few slices of pineapple? This is because pineapple contains a unique enzymatic protein called bromelain that actually digests other proteins. So as you eat pineapple, it's literally breaking down some of the proteins in your mouth. This is why some call pineapple the fruit that eats you back. So what I would look at if I were reading this is I would say, well, they said that there's a enzyme called bromelain that it's a protein, it's an enzymatic protein, and they said it actually literally breaks down proteins in my mouth. So what I would have gotten out of that is proteins, or rather, I should write down enzymes can break down proteins. And we know in the example of ribosomes that ribosomes, that looks messy, build proteins. It's the first page of work. So enzymes can both build and break down. And your book calls it when it breaks down stuff, when there's a process of breaking down biomolecules, eating, right? Your book calls it catalysis. Catalysis. Under the big, broad term of metabolism. When someone tells you what your metabolism is high, what they're saying is you're breaking down stuff quickly and you're building things quickly. When you're building something, instead of breaking down, but building, putting things together, catalysis the process is anabolic. So there's two sides of, that's what the book calls metabolism. So you can break things down and build things up, but in the case of a good example of building something is ribosomes. One of the many organelles you should know. It is not a membrane-bound organelle, so it's found in both prokaryotes and in eukaryotes. Ribosomes are RNA, they're actually made of RNA that can build proteins. But this bromelain is an example of a protein that breaks down protein. So what am I doing in this? This is a modified version of Cornell Notes, What I'm doing is I'm processing the information. I cannot expect that I'm going to read something and answer it, and then that's all I have to do. If that's my expectation, then I'm going to be sadly mistaken, and I'm not going to do well in high school, especially in a challenging high school. What you need to expect to do is take all the different sources of information that you have, like your book and this activity and the class notes and other activities, and put it together. In class, some other information I gave you. In class, I said enzymes have 3 very specific and important qualities. I said 1, they can be made of proteins or RNA, and there's 2 examples right there. 2, they are not used up in the reaction. So if you have, when you're at the beginning, you have, I'm just going to write the word enzyme, plus you add the substrate, you end up with the enzyme, because the enzyme is not used up, plus the products, whatever that product is. It could be more than one, so I'm going to go ahead and put products. And substrate could be more than one substrate, so we'll say substrates. So the only things that's changing in this reaction is the substrate is changing into products. That's the reaction. This guy here, this enzyme, does not change. It's not used up. If I start with 50 milligrams of enzyme, I end with 50 milligrams of enzyme. It's still there, not used up in the reaction. The substrate will be used up or will be made. The product could be making something. It doesn't have to be breaking it down. Remember, it could be catalysis or it could be anabolic. So your body creates lots of different proteins. They carry out critical functions in your body. Proteins are made of hundreds of thousands of smaller units of monomers called amino acids. Now, everybody in this room should know that. That should be a really big review for this final. This should not be a surprise if you see an amino acid in somewhere. I'm not going to go ahead and draw it again and go over it, but I'll draw it really quickly, I guess. But I don't want to rehash everything. We don't have time. This is something you should have been doing now for a month because you've known this is coming. So you have an amine group. It's a central carbon, a hydrogen. There's some kind of R group. Remember, this is a variable. And then that's connected to C double bond O, OH. This is an amino acid. There's 20 of them, 20 different amino acids. They all have different qualities. They're bonded through peptide bonds in long chains. This is the monomer called an amino acid. The structure of proteins determines the function. They function in living systems. The table below highlights some key functions of proteins. So let's be clear. When they say structure, when they say the word structure, just as a note, let me make a note here. Let me highlight this. If I were reading again, if I were a student reading this, I would ask the question, what do they mean when they say structure? What do they mean? Well, I hope that at this point you understand that when they say structure, they mean shape. The shape of the protein. Now, a protein shape, it's going to twist and it's going to turn and it's going to fold. It folds over and over again. And we talked about this. And when remember, this is a chain of amino acids. They're chains. They're not, when I look at this, it's a block, right? But is it a block? Is it a single block? Really? What is this, really? Right? It's a long chain that's folded, that's wrapped around, it's tape, right? And tape is a long ribbon that's wrapped around a core, correct? So when you're looking at a shape of a protein, really what it is a long ribbon of a bunch of amino acids all bonded one to the next. And they form a shape. The diagram in your book, in your handout, in your workbook, has a ribbon, and what it has is maybe part of the ribbon might be red because it's hydrophilic, so it loves water. These are the amino acids that love water. And maybe part of it are hydrophobic. These guys don't like water. Or maybe they're acidic. Or maybe they have some kind of quality that makes them interact with each other in something that's called hydrogen bonds. Remember those? They're weak bonds. They're the things that hold DNA together. They give DNA this shape. And that's what these are going to represent here. Especially ladies in the room should know this. When you're putting your hair in a bun, you use a rubber band, use a headband, you twist things together, you use a clip, right? A young lady yesterday asked me for a paper clip to clip their hair down. One of those binder clips I have to clip their hair down. So these act as clips. They hold this protein shape. These hydrogen bonds act as clips. And so there are these, and when you start to put all these hydrogen bonds together, there's other kinds of forces like London dispersion forces and all kinds of things that are going on. But these are all bonds, weak bonds, very weak bonds. So this protein has a shape. And if the substrate, in the case of an enzyme, one of the functions of a protein is being an enzyme. Another function of proteins we discussed earlier is to be a signal molecule. Or a receptor. So they could be a signal molecule receptor. As we discussed earlier, insulin has a shape. Let's just make a pretend shape here. And the receptor for insulin is going to have a shape. They're going to fit in each other. These are very simplified shapes. Obviously, the real shapes you see in the book are very complicated and they're hard to see. So we draw these very simply. These have their shape because of what? Hydrogen bonds. It's the only reason they have their shape, because of hydrogen bonds. So this fits into that, and that starts a whole signal. So those are some of the proteins. So what I would have written after I've thought about all this, where did I get all this information? From the class notes when I drew the 1,000,001 proteins in class, when I discussed hydrogen bonding, from your textbook, from the reading in this packet talks about hydrogen bonds as well. So let's call this then say shape determines function. Also, shape is determined by hydrogen bonds. For the most part. Weak bonds. What about messengers? Same thing with messengers, right? Same thing with your skin. Your skin is made of cells who have proteins that allow them to lay flat and stick closely to one another. That's good if you have skin, correct? Would it help you if your body's, to be skin, if your cells of your skin were this way? Not as much. The shape of the cell also determines function. In your intestines, your cells look like this. And then the next cell is they're welded together with these little tight junctions. Why do you think that this and all the food is here? All the food you're digesting is here. Why did your cell decide to, why are the cells taking on this shape? What does this do? How does this shape apply to function? What's the function? What function are we talking about? Why does the folding help? Think. If I was going to absorb something, do I want to have a bunch of stuff like this, all these folds like this? Or do I just want to have a flat surface? Which would be better for folding, for absorbing? Anybody have a guess? Look at it. By folding it up, I have more surface area with the same volume. So I have a sheet of paper, takes up this much space. Do you see it? If I had a cell with this much, to have this much surface area, I have to have a cell that's this big. Correct? That's a big cell. But what if I don't want something that's that big? What if I take this and fold it back and forth, over and over and over and over and over and over again? Now it's folded. I have the same surface area. You see it's there, right? But now it's folded. Now I have less space, less volume, so my cell can be this thin, right? But I have the same surface area for absorption. So things can still get absorbed in these spaces, but I'm taking up less space volume-wise. The cell can be smaller and have the same surface area. Is that clear? Yes or no? Please help me out. So the increase by folding, you increase surface area while keeping the volume of the cell small. So my surface area increases while keeping the volume small. So now I can diffuse quickly. As soon as I bring the food into here, it can go quickly here. If the cell was bigger, it would take longer for diffusion to happen, and my digestion would be much slower. So I want to be efficient, so folding helps. Your brain. We used to think that if you were smart, you had a bigger brain, because we could look at some of our other primates, and we could think about our brains, compare our brains, we see our brains are bigger. But we find that this is not true. What we find is that what equates to intelligence is not just the size, but how many folds you have in the brain. What is folding doing? What's all this folding doing? Increasing the surface area. I'm increasing the space without increasing the volume of the skull. I'm increasing how many connections I can make, because I can have more cells in the same space by having folding of the tissue. So folding is a very nice trick that life uses to increase how much you can fit in a small space. Which explains why some animals are very small and we get very smart. And we have very large animals that are just kind of like, if somebody ever calls you a smooth-brained individual, they've just insulted you, right? They're saying you have low intelligence. Obviously, the sketch is not accurate, so don't take that your brain looks like that. It doesn't. You can look it up. Your brain does have folds. It just doesn't look like that. Another thing is structural component. Again, we just talked about enzymes, but if you look at proteins that are if you're making hair, you want to have proteins that are going to all intertangle one another, right? You want hair to make strands, correct? You want nice tight weaves, right? And that's what you have with a strand of hair. Pluck a strand of hair out, you look at it, it looks like one thing. It's actually a bunch of interwound proteins. If you're making fingernail, you want something that's stiff, that lines up nice and straight and solidifies and is something solid and hard, right? So your fingernails are made of proteins that stick together, but they're very stiff. So their shape and structure determine their function. It would be really suck if your hair was like your fingernails, or if your fingernails were like hair. They wouldn't work for what you're trying to do. And of course, transport and storage. All right. One great example is hemoglobin, which we've talked about in class, has four domains, 4 polypeptides, 2 copies of an alpha polypeptides that are exactly the same, and we'll talk more about these when we do genetics, and 2 copies of another protein. So they're both called hemoglobin, alpha hemoglobin, and 4 chains all make one molecule. Obviously, again, this is a sketch. This is called hemoglobin. The whole protein is called, even though there's 4 polypeptides, and in the center of this, you have this heme group that's basically an iron plus 2. And what that does is you get oxygen coming in, O2. What's going to happen? What happens with iron and oxygen? Oxygen rusts iron, right? It doesn't quite rust, because rust is a stronger bond. Hemoglobin keeps the two associated. The iron grabs onto the oxygen, but it doesn't completely bond to the oxygen. Yes. So, but in any case, it is why your blood is red, because what color does iron turn? Red. When the oxygen is present, That's why your blood is red when there's oxygen present, because the iron changes color. But hemoglobin is in a red blood cell. So that's an example of proteins acting as a carrier. What are they carrying? Oxygen. In the middle of red blood cells that have this shape, if they're normal functions, and inside you have a bunch of these hemoglobin molecules, and I mean a bunch. The whole cell is full of them. It gets rid of the red blood cell, even gets rid of its nucleus. Because it gets rid of its nucleus and all the other organelles, it really is literally just a sack of hemoglobin, and this red blood cell only lives 3 months. After 3 months, it dies. You replace your red blood cells every 3 months. So this red blood cell has a mature red blood cell is I don't even know what just happened. A mature red blood cell is sorry, I'm trying to do two things at once. That's never a good idea. I keep telling you not to do it. So mature red blood cell lasts 3 months, replaced every 3 months, and is a really good indicator of a lot of your biological functions. When the diabetic checks what's called their A1C, all they're doing is seeing how much sugar is on the outside, is stuck on the outside of the red blood cell every 3 months, because sugar sticks to the outside of the red blood cell. All right, so you take a look at enzymes, catalysts. They speed up reaction, right? So again, I'm not going to go through this whole thing because we don't have time. I would love to go through the whole thing with you, but we don't have time. So the catalyst, that means you're speeding up the reaction. It lowers the activation energy. Enzymes lower activation energy by attracting the substrate and active site to its active site. People are like, what is the active site? What is an enzyme? Well, there you have it. Enzymes, enzymes can break down molecules, or they can build them, as we said earlier in the previous page. All right, so you guys can take more notes on that if you'd like. When we look here in the model, they give you a model. They tell you what's going on. You should pay attention. Enzymes are active in all cells as well as in the digestive system. Lactase is found in the digestive system. Now let's go ahead and highlight lactase. The word lactase, it says ASE. What does that make this? It's an enzyme. So lactase, it says lactase breaks down lactose. Which one of these, the blue or the purple? Which is being broken down? The purple. These are three pictures of the same thing. This is the beginning, this is the middle, this is the end. In the beginning, you have two things. In the middle, they become one thing. And in the end, one of the two things that you started with is broken down. Enzymes start and end the same, correct? So which of the two is an enzyme? Which of the two is an enzyme? The blue. And what is it? Lactase. So what does lactase act on? By the way, everything's in the names. Lactose. Oses means sugar. Ace means enzyme. Lact means a sugar that is made of two it's called a disaccharide. We've talked about this. A disaccharide is made of two monomers. One is glucose and the other is galactose. We literally talked about this when we did carbohydrates. So one of these, it doesn't matter which one you want to label, is glucose, and the other is galactose. So it says represents a reaction in which lactose is broken down into by the enzyme lactase. So label lactose, already labeled, check. Lactase, check. Active site, the active site is where on the enzyme stuff's happening. There it is, active site. Check. Enzyme substrate complex. That's when you have the enzyme and the substrate together. Where is that? Right here. I'm going to continue this all day, so you'll have almost all the answers posted tonight. All right, so now I've identified here the lactose, where the lactose was, where lactase is, and where the active site is. I've also identified the enzyme substrate complex. I think that's all fairly easy. I don't think anyone would argue that it is not easy. The products of the reaction, I would hope that at this point you see that the products are glucose and galactose. So now to review again what we just did so that we're all clear. So we looked at the three definitions, the 3 points. If you missed it, just rewind the video a little bit and look at it. But what we know here is that enzymes are not changed in a reaction. So what I start with, I end with. The substrate can be changed. It can be added to or broken apart, one of the two. The enzyme is going to make all the enzyme does is make the reaction go faster. It doesn't actually get involved in the reaction. The example I used in class is that if somebody hooks you and someone up, they're not part of the relationship, they just introduced you. So an enzyme is just introducing the conditions that allow the reaction to occur. So the lactase, and again, please rewind and watch the video because I think we did a decent job explaining how enzymes work and why they work. But in any case, what we have here is an enzyme. Which of these is the enzyme? If we start with this, we have this in the middle, and we end with this. One of the two got broken down. The purple one got broken down. Since the purple one got broken down, the purple one can't be the enzyme because the enzyme doesn't change in the reaction. The enzyme stays the same. It's the enzyme you start with, you end up with. So in the beginning, you have the substrate, which in this case, if you read the problem, is lactose. It says sugar. It's a disaccharide. It's made of two glucose and a galactose, two sugars. And these two are linked together, and this enzyme creates, in its active site, creates the environment that allows these two to break apart very easily. And therefore, we require less energy to break these two up. They're broken up. Glucose and galactose separate. So these are the products. This is your substrate. This is your enzyme. This is your active site. In between the beginning and the end, you have the reaction happening inside the active site. So this is called the enzyme substrate complex. Enzyme, substrate. Enzyme, substrate. Using the names of the reactants and products instead of chemical formulas, create a chemical equation for the reaction catalyzed in Model 1. Well, then that would be lactose plus lactase yields glucose plus galactose plus lactase, because lactase did not change because it is an enzyme. That ends that page. Now, referring to the chemical reaction, you wrote in the question above, is lactase a reactant, a product, or neither? Well, I would hope at this point you'd know it's a reactant. It's not a product. You know it's not a product. And of course, it's not going to be neither. So in any chemical reaction, you have reactant. It could be more than one reactant, so I'm going to put the S in parentheses. You have the yield sign, which means something happens in between, and then you have some product. It's going to change. The bonds are going to, and it could be more than one product, so I'm going to put an S. So your substrate is at the beginning, lactase. Oh, well, I misread that. I read that as lactose, so that's not right. It's not the reactant. It's not the product. My bad. I misread that. It says, is lactase. Lactase is the enzyme. And as I said earlier, enzymes end up the way they begin. They're just matchmaking. They're not getting involved in the reaction. So it's neither. It's not a reactant or a product. It is just the thing that's hooking up the reactants together to create a product. It lowers the activation energy. Describe the role the enzyme lactase played in the reaction. It creates an environment that allows the product to form with less energy. And this thing jammed. Requiring you to listen to what's going on. I'm giving you the answers, but you don't have to. You can do it. Again, there's several different things you can be doing. We only got a couple days left, but you can't be talking. You guys can write little messages to each other all day long. But at this point, I just need to get through this. Minutes? Huh? Page 20 is a blank page, so you don't have to worry about it. What do you think could happen? This is page like 4 or 5. You can. What do you think could happen to the end? What do you think could happen to the enzyme lactase in the end of the reaction? Nothing is possible, but what could happen is another lactose, and what probably will happen if there's enough. Another lactose, it is lactose we're dealing with, right? Yeah. Lactose could find the active site again. If it finds the active site, then another product is going to be formed, and it's just going to keep going and over and over and over and over and over again. Yeah. Between what? So lactose is a substrate. Anything. So OSC is a sugar. If I don't want to rewind, or go back. So let me just draw it out here. Lactose is a sugar that's made of two monomers, two carbohydrates. One, I believe, one's I don't remember what galactose looks like. I don't remember if it's a hexose or it's a pentose, but one's galactose and the other is glucose, and they're bonded together, right? And through dehydration synthesis. And what happens is that there's an enzyme called lactase. ASE means enzyme. That's the difference. And what happens is that this fits into this well enough. I guess I have to erase this a little bit. This fits into this well enough that it creates into this part here is called the active site. This part of the enzyme right here is called the active site, where the substrate's going to bind. It's called the active site. And the substrate, in this case, is this thing here, by the way, just to be clear, these two together is what's called lactose. This is the sugar that your mother fed you from her breast. This is the sugar that all mammals make in their mammary glands. That's true. Not everyone breastfed, but most people are drinking lactose regardless. So, since formulas usually have lactose in them, but it's a good point. So the lactase, they bind, and the lactase creates an environment here that lets these two split up. So you end up with two sugars instead of one. That could happen, and then another one can come and bind, and another one. But the one that always stays the same is lactase. Lactase doesn't change because it's not part of the reaction. One thing turns into two things, one thing turns into two things repeatedly. You get more and more glucose and galactose and less lactose. So if you have a glass of milk, you're going to end up with less and less milk. You girls are going to keep talking until I say something, huh? So you have less and less milk and more sugar water. So this is gonna quickly turn into, if you have lactase in there, a clear liquid with a bunch of glucose and galactose. You wouldn't be able to tell. Again, I don't remember if galactose is a pentose or it's 5-sided or 6-sided, but in any case, it'll be clear. Because milk is white only because you have lactose in it. There's proteins in it as well. But as this lactose gets broken down, it'll become clearer and clearer. Proteins and fats as well. There's also fats in whole milk, as well as some proteins. Enzymes play a very specific role in cells and will work only on specific substrates. For example, while lactase breaks down lactose, lipase is an enzyme that breaks down lipids. So if you've been following directions and you're doing Cornell notes, then certainly on the side, we would be taking some key points here that lipase is an enzyme that breaks down lipids, while lactase breaks down lactose. And the key point that I would write down is that enzymes are very specific. If I were you, I would be thinking, Mr. Mendoza doesn't want me to memorize lactase and lipase, even though it's in the name. Lipase breaks down lipids, and lactase breaks down lactose. Even though it's in the name, I don't think Mendoza's going to expect me to memorize these two things. Using lipase and lactase as examples, describe most enzymes, how most enzymes are named. The beginning of the name is what the enzyme works on. ASE is the suffix that means enzyme. So the beginning of the name is what enzymes work on. And so when you have lipase, it works on lipids, and lactase, it works on lactose. Suppose the image below is a model of a different disaccharide, sucrose. Would lactase be able to break down sucrose? Explain the answer in terms of the model. Okay. The answer, what answer do you think you would give? Would the enzyme lactase be able to break down sucrose? You said no, and that's correct. The answer is no. No. Why not? That's right. It does break down only glucose because the enzymes are very specific. But the reason is, and you have to go back and watch the beginning of this video to really understand what we mean by the shape. The reason is the sucrose won't fit in the active site. So if lactose, as you saw earlier, if this is lactase, lactose fits in it no problem. Oh, great. Now it's connected for some reason. If this is lactose fits in. Everybody's happy. But sucrose would have a completely different shape. And it wouldn't fit. It just wouldn't fit. And because it wouldn't fit, lactase can't work on sucrose. You would need sucrase or something like it. You need an enzyme that's made specifically to break down sucrose. So lactase only breaks down lactose. You have an enzyme for every different substrate. I have to give you a little bit of a but. And this is always in biology, there's always these rules, and then we have these exceptions. Enzymes can match if the two substrates are close to one another, if they're almost there. So For instance, do you agree this is round? Yeah. So this is not round, but it still fits, right? This would not fit. Not only is it too big, but it's the wrong shape. But this is small enough, it fits in the active site, it can work. So you can get close. Some substrates get close enough, but it's rare, and the substrates have to look a lot alike. And we can take advantage of that if we're making poisons. When we make poisons and weapons of war, we take advantage of that. That we can make something that looks a lot like the substrate, but then doesn't act the same way and ends up killing someone. I have to say that I made a big error when I said this. This is not going to, milk is not white because of the sugar. It is white because of fats. It's not going to be none of this is going to be on a test, but I just want to make sure I'm not giving you wrong information. Milk is white because of proteins and fats. They reflect all the light, and so it's white. It would not become clear if you added lactase, but it would break down the sugar. Is that clear? Does that cause a problem for anyone? All right. So I do apologize for that. White milk would not turn clear. This would still be white. I wish I could. This would still be white. It's just the sugar would be broken down. And again, I did look it up, and galactose is a hexose. It's just a structure. It's just connected differently. So it'd be two hexoses, and it would be no longer would it be these two hexoses linked together. They would be separated. Does that make sense? So I just want to make sure I correct that, because I always hate giving bad information to people, which is going to happen because I make mistakes, but I'm really try to minimize that. So this would actually be more something like sorry, I just want to make sure I'm accurate before I upload the video and everybody's clear. All right, thank you. All right, let's take a look at Model 2. Changes in the environmental factors such as pH. Now, this is incredibly important because I can almost guarantee you're going to have a question like this on the test next week. When we look at pH, we look at pH and increases in temperature. Those are two things, and I have to say I'm going to add salt concentration. So, but usually it'll be pH, how acid, so then again, what is pH? Acid, base, neutral. Running from 7, which is neutral, to 14, to 0, neutral, acid, base. Okay, how much hydrogen ions do you have available? That's acidity. 0 to 7 is acid. And 7 to 14 is base. Let me use different colors. So 0 to 7 is acid, and from 7 to 14 is base, 7 is neutral. All right, because the pH of 7 is neutral. It's not acid or base. So that's acidity. That can impact the shape of the protein. And then again, I would ask you to rewind and you would understand that a protein shape is due to hydrogen bonds. And hydrogen bonds, and these little green things are hydrogen bonds. And these hydrogen bonds, and we talked about this before, we saw this in DNA. DNA has a shape because of hydrogen bonds. And these are all made of amino acids, and the amino acids have R groups, and the R groups can make hydrogen bonds. And different areas might have different properties. For instance, these R groups here on these amino acids might be hydrophobic. These out here might be hydrophilic. And then we might come over here, maybe these are acidic, or maybe they're also hydrophobic. And over here they're acidic, so they're kind of, well, they are hydrophilic, but they're also acidic. And maybe over here they're just hydrophilic again, and here they're hydrophilic, but over here they're hydrophobic again. So they have different, that makes shapes. And why do they have this shape? Look, if you take hair and you braid it, and you swoosh it together and tie it all up and add clips, you can make hair turn into different shapes, correct? You can look around the room right now and see a bunch of different people with a bunch of different shaped hair because of all the different things that they've used to tie them up, to wrap them up, to hold them together. Correct? If you don't hold them together, what happens to hair? It goes crazy. It loses all its shape, right? It goes wherever it wants. You have to hold it together. Do you agree? We use all kinds of products. I have oil in my hair. What do they call it? I forget the name of the oil. Oil or something like that. What she said, yeah. And you can use gel in your hair, use hairspray, right? But you have to hold it together. If you don't hold it together, it just goes nuts, right? It's the same with a protein. It's just, the reason it has its shape is because of these hydrogen bonds. These are the clips that hold it to shape together. If you add acid, you're adding hydrogen ions. If it gets acidic or too basic, then you're removing or adding hydrogen ions, then that changes the shape because it interrupts hydrogen ions, either more or less, more hydrogen ions or less hydrogen ions can change the hydrogen bonding. And what that does is it changes the shape of the protein. Well, that's bad news for the enzyme because the shape determines it won't fit anymore. If you change the enzyme shape enough, it doesn't fit anymore. The substrate doesn't fit, so it's not going to work. Then the other thing is that you can add heat. You can add fire. You can add heat. And by adding heat, what that's going to do is it's going to make this move around a lot. And what will happen is the increased kinetic energy is enough to break the hydrogen bonds. It's not enough to break the peptide bonds that hold the strand together, but it is enough so these bonds stay together, but these green hydrogen bonds, did you just erase everything? So these bonds here hold together, so the amino acid keeps its primary structure, so it's all right. but its shape changes because the hydrogen bonds are broken. A really great example of this happening is water. If you go get your hair done, I don't know what girls call it now, but they used to call it getting a perm. Some guys would get perms. You get your hair done, you spend a lot of money, a few hours getting your hair done, and then you go out in the rain. What happens? Everything's done. All that hard work for nothing, because what happens is you're disrupting what you're doing, what your hairstylist is doing when you're sitting there for hours, is you're creating a shape, and then you're locking it together with what? What are you using to lock your hair together? Chemicals. But the chemicals are doing what? What are they creating? They're using heat to break the hydrogen bonds you have now. So you use heat. If you use enough heat, you can break the hydrogen bonds your hair naturally has, and what happens to your hair? It straightens. So it loses its shape, right? Its natural shape is lost because you straighten your hair because you've added just enough heat to break the hydrogen bonds. Does that make sense? Now, if you want a different shape, then you can put them in curlers, right? In a curlers, you see women in curlers? Have you ever seen that? Those little rolls that they wrap their hair around? Yes or no? Yeah. Okay. So you can wrap it around a certain shape and then add some chemicals that do what? What are you creating? A perm. But what are you creating? What kind of bonds? Hydrogen bonds. And then you release, once they set, you take the curler, the shape out, the plastic shape, the little rollers or whatever, take them out of your hair, and it keeps the shape for a long time. Unless you get wet or it gets hot again. So the worst thing for hair that you have you're holding together with hydrogen bonds is hot, humid air, right? What happens to your hair? Bing! Frizz. Everybody's hair does the same thing because these hydrogen bonds are broken. So enzymes do the same thing. With enough heat, pH, or salt will do it too. Why? Because salt is ionic. It'll break the hydrogen bonds. So bad news. So if you lose the shape, there's an example. You saw the shape before. Now you see it's not going to fit, is it? The shape's all wrecked. That's exactly what happens to your hair. Because you've added heat, the shape of the enzymes changed, and the substrate doesn't fit in the active site anymore. We call that denatured. Please know that word. Denatured. It's lost its natural shape. It's lost its natural shape. Denatured. Very important word. If you've denatured the protein, it won't work anymore. Now, what can you do to make it go back to its original shape? Well, it's the same thing you do with your hair. Just let it relax. They even have I think, and girls, correct me if I'm wrong, maybe some guys know this because I know my mother was a hairstylist, and so I knew a lot of stuff about, at the time, words that they would use. But they used to call it relaxer. Do they still have that? So you can add relaxer, and what that does is it just relaxes your hair, goes back to its natural shape. It loses it loses the hydrogen bonds, and everything goes back to normal. It straightens hair, but it breaks the bonds that you made when you do a perm, for instance. You've created those hydrogen bonds. You add relaxer. What are you doing? You're breaking it down, letting it happen, and then you dry off your hair, wash it, dry it off, and now leave it alone. What's going to happen to the hair? It's going to go to its natural shape, right? Right, but people that have perms, they want to get rid of their perms, they use relaxer. So it's just about breaking the hydrogen bonds. And then if you wash your hair off with the relaxer and then let your hair alone, the hydrogen bonds will reform. They'll go back to its natural shape. That's my point. Does everybody agree? Thank you for the clarification, though. I appreciate it. Everybody get that? So that's the whole point. using your new knowledge now, you should be able to describe why temperature changes and impairs the enzyme's ability to catalyze reactions. Again, the shape, if you change, well, let me rephrase that. If you add enough heat, hydrogen ions, or salt, you break the hydrogen bonds, which makes the proteins shape change. And the substrate won't fit. So the enzyme does not work. I know it's a lot of words, but I just want to be clear. So your enzyme loses shape, it loses function. Now, when you cool it off, you do the reverse, right? You add more hydrogen ions, and it loses shape. If you remove it, it's fine. So by the way, that's why if you have an extremely high fever, you stop working and you come close to death, by the way. What happens is, you go in the hospital, what are they going to do? If you have extremely high fever and you're dying, or your baby is extremely high fever and you're freaked out and it's crying, and it's just your baby's just laying there like this. You have 105 fever. What are you going to do? Take it to the hospital. What are they going to do when you take it to the hospital? Do what? Cold. Yeah. Get it cold. Cool that baby down, because then the hydrogen bonds can reform and the enzymes can start working again. Because right now it's so hot, your baby's so hot that the enzymes have lost their shape. They're not working. The hemoglobin's not holding on to oxygen. You're not breaking down glucose. Everything's stopping. You're in an emergency situation. You got to cool that kid down, or that human, or that adult. We used to just use an ice bath, but now we have different machines that pretty much they'll even take cold saline solution and put it in your blood to cool you off even faster. So they need to cool you off to get those enzymes back to their original shape. You have to put that relaxer in to get rid of the perm. You have to put that, you have to add that, you have to do the reverse of whatever made you lose that enzymatic shape. Because everything that happens in your body happens because of enzymes. All right, so hopefully you have some questions after all that. So with cellular enzyme and cellular reaction simulations, when we get back, we'll do this experiment. I kind of wanted to do this one instead of the one that I chose. You don't have to be, you can do this actually on your own if you want, because look at it, if you think about it, all you need is some 2 bins, right? 2 boxes, large containers, 100 assembled pop beads or toothpick pairs. So you can choose toothpicks, which almost everybody can get a hold of pretty easily. And those toothpick pairs are going to be your you're going to have 100 of them, and they're going to be your lactose. So when you do this coming back from break, your lactose is going to be these pairs of toothpicks where one is glucose and the other one is galactose. All right, so when you're doing this experiment, if you decide to do it at home, if you do it and then you can just use a timer, paper clip the paper and pencil, record your data, and bandage tape, because obviously you're going to tape them together, right? So that's what happens with to make lactose, glucose and galactose through dehydration synthesis are bonded together, and the tape you're going to use is going to model the bonding. And then it tells you to do this one step at a time. You have a warm-up investigation. You go ahead and you consider the models. How long does it take you to assemble and disassemble? And then it tells you to do some other activities here and some other activities. And here again, we're talking about hydrolysis and dehydration synthesis. So if you read this and you saw that it's talking about enzymes and it mentions lactose molecules disassembling, and it uses the word hydrolysis. So there's two words that we've covered that are going to be on the test likely: hydrolysis, and dehydration synthesis. They're opposite one another. Remembering lysis means to break up. Hydro means to use water, so you're using water to break up something, usually a pair of something or a polymer of something. And dehydration again has to do with water. You're taking water out in order to synthesize something. You're making something. So when you're making, you're doing dehydration synthesis. When you're taking apart, you're doing hydrolysis. And this kind of models that for you. Then you have some analysis questions using data collected. Calculate the rate of production per minute without the enzyme, and then how much with the enzyme. They want you to show your work. What is the overall effect of the enzyme on the lactose hydrolysis, the breaking up of lactose into galactose and glucose? And what is the purpose of the no enzyme condition in the experimental design? Now, if you're not doing this stuff, even though it wasn't assigned, it's going to be harder for you to do the experimental design section that's due on the day of the final, which is for you on Monday. So when you're looking at this, if you think about it, if you have a graph, someone close the door, please. If you have a graph and here's energy, and this is time. This is the rate of react. This is time, and so as you move along, if you measured this, what you would see is you'd start off with your products, you'd add some energy, and then you'd have the reaction occur. And you'd end up with the products here, and here you'd end up, these were your reactants. That's without the enzyme. With the enzyme, you end up with the same products, but it takes less energy. Everybody should recognize that. That shouldn't be. And because it takes less energy, You're not increasing the temperature, so the amount of energy available is the same. If you did a different graph that looked at how much product you were making over time. So in this case, it would be glucose plus galactose. How much you'd be, at first you'd start with 0, right? There wouldn't be any. And then as the reaction actually started, right about here, the reaction would start. If you're using an enzyme, it would start, it would need this much energy. But because it needs less energy, without the enzyme, you'd have something like, lactose would just pretty much stay at 0. With the enzyme, all of a sudden, you start to see an increase, and your rate of change, your rate is much faster than the rate of without the enzyme. So you produce a lot less product without the enzyme in the same amount of time. How do you tell rate? Who can tell me how you calculate rate of reaction? You should all know this. Everyone in the room should know how to calculate rate. You're about to take a math and science final, so you should know that. Anybody? Scary. Very scary. So a rate of reaction is the slope. You know how to calculate slope, do you not? Slope of a line is change. The change. All right, slope of a line equals the change. Well, what is the change here? How much product over time? Think about what it is. How do you calculate slope? What is it? I can't hear you. No, that's y equals mx plus b is the equation for a line. And in y equals mx plus b, m is the slope. The y-intercept is b. Then your x and y are points on the line. So that's not how you calculate slope. How do you calculate slope? And I said it's not. That's the equation of a line. Do you need me for something? Yeah. I'm asking you, how do you calculate slope? Yep. Which is on top? Change in y over change in x? Is it? Why are people not looking it up? That's really my question. Oh, you did? If you're sitting there and you're like, I'm not sure what slope is. So if I were a student and I were worried about participation, making things happen, I would be like, let me go ahead and look it up. How do I calculate slope? Changing y over changing x equals product over time. Changing y, how much product do I produce? By the way, the clue was right here in front of you. You didn't have to look it up. I wrote down here, what did I write down? The product, which is what? On what axis? On the y-axis, over time, which is on the x-axis. So changing y over changing x gives you the slope of the line So how do you calculate that? You pick 2 points, any 2 points, 1, 2, and you say it doesn't matter as long as you're consistent. You call this point 1, call this point 2. So it'd be y2, whatever y. How do I find y2? I go across minus y1. I go across divided by x2. I go down, and x2 is over here. So I go, and then x1 is there. That gives me the slope of the line. That tells me how fast things are, how much product I'm making in any given amount of time. That's the rate of production. Rate of production. Rate means what is the change over time? Change of whatever over time. In the case of you driving, that means speed. Distance, change in distance over change in time, right? I'm going 50 miles every hour. That's how much change in distance over change in time. Does that make sense? So if you're ever asked for a change, a rate, if you look at this, you don't even have to do all this math, really, except you do if when we do the experiment, you will. But when you look at it, which one's changing faster? Ouch. Which one's changing faster? Is it line A? Oh. Forget it. So line A or line B, which line is changing faster? Which is making more product in a given amount of time? B. Its slope is higher. It's a bigger angle. So you can just look at it and tell which one. But if you want numbers, you have to use this formula. This is how you calculate slope, change in y over change in x. And they have you graph it, of course, so that you can do your math, calculate the slope. And then they ask you some more questions here. Assuming the bin were big enough, what would the results be if 10 enzymes were added? They want you to see what is it possible, how can you make it go faster? Adding, so how can you, the question they're answering here is how do you affect the rate of reaction? How do you affect how fast you're making product? Well, you can do a bunch of things. You can increase the temperature. Up to a point, you can increase the temperature, and it would speed up the reaction. After a certain point, though, it would stop working. And you should know that because of that. Well, you haven't watched the video yet, so you don't know. You could increase the amount of enzyme. But that's assuming you have enough product. I mean, not product, reactant. All right. So what limits the reaction? What's going to stop it from going faster and faster and faster and faster? Well, how much reactant you have. That's one. And two, how much enzyme? All All right. This is page 13. This is all pre the work that you were supposed to do. So all these we'll talk about later. Here it says introduction to catalase, and it says it really is important that you look at this. This is that ribbon model that I talked to you about. And this is the enzyme catalase. I talked to you about shapes. I said, look, enzymes have these shapes, and the shape of the substrate has to fit the shape of the enzyme, this being the enzyme. The enzyme is going to create an environment in which the substrate or substrates can complete the reaction, the change. So your substrates here now you can have more than one substrate. You can have two things. The enzyme can fit two or three things in it. It depends on the enzyme, depends on the reaction. But what the enzyme does is it creates an environment where the substrate can become the product. So it changes and becomes a product, like any good chemical reaction, where the substrate is a reactant. Start with reactant, end with product, just like the graph we just looked at. The enzyme does not change. You started with an enzyme, it ends with an enzyme. The enzyme's active site will match up with the substrate, and it's here. This is the active site. That active site doesn't change before or after. Nothing changes with the enzyme before or after. This is the active site. The active site's just ready for taking in another substrate. But these are just drawings, sketches to make a point. This is what they actually look like when drawn with their secondary shapes, their hydrogen bonding, all the little hydrogen bonds making the amino acids, pulling the amino acids towards each other. I keep using hair as an example. I mean it. Hair is very similar. And if you want to straighten your hair, and your hair is curly like it is here, and you want to straighten it, you have to add heat or chemicals that are going to break the hydrogen bonds. And if you break the hydrogen bonds but not the peptide bonds, then the hair becomes straight. Hair is a protein. But it loses its natural shape. When you lose its natural shape, that means that, going back to the model here, that means that if you change this shape, it doesn't fit the substrate anymore. That means it won't work the way it's supposed to work. Unlike hair, which it's still going to work, right? It's still hair, whether it's curly or straight. With an enzyme, it doesn't work anymore. So if you change the shape by increasing the heat too much, or increasing the pH, or decreasing the pH, or increasing the salt concentration, then you could end up messing up the enzyme, and the enzyme loses its shape. It doesn't work anymore, and you end up dying because the chemical reaction doesn't work. A good example of this is having a high fever. If you have a really high fever, your enzymes start to unravel. They start to lose their natural shape. If that happens, you're dying. So when you go to the hospital, they cool you off. They put you in an ice bath, or they put you in a machine that cools off your blood. They'll stick you with an IV with cold water. They got to do something to bring down your temperature so that the heat allows the enzyme to go back to its natural shape. This happens to hair all the time. People put in hair relaxer, right? Another word, hair straightener. And all it does is it breaks up the hydrogen bonds, and your hair becomes looser, and it could even get straight, right? They form it the way they want to form it. They'll put curls in so that their curls are looser, right? And some people call that a perm, right? But then it gets a hot, humid day and they go outside and it's humid or it rains. What happens to their hair? It goes right back to its natural shape, right? Because you broke the hydrogen bonds that you made. So by putting in relaxer, you're breaking hydrogen bonds. When you put your hair in curling irons or whatever, and you let it set, now the hydrogen bonds are reforming the way you want them to reform, so they have the shape you want, right? And then you walk out and your hair stays like that all day, unless it's humid or hot or wet. Then what happens is hydrogen bonds break, and then they go back to their natural shape. Does that make sense? So it happens with hair all the time. If you want to think about how heat affects protein, think about curly hair. What happens when people iron their hair? It breaks all the hydrogen bonds. It doesn't break the protein, right? It doesn't break up unless they do too much. My brother did that to himself. He added too much hair straightener and it broke into pieces. He was crying for 2 days. I was like, well, you should let it alone. Why are you messing with it? But anyways, so when we look at catalase, the catalase is the enzyme you're going to be working with on Monday. Hopefully, if it comes in. I ordered it. Will it come in on time? I don't know. If not, we'll have to punt. We just won't do the lab. I don't know what else to do. Okay. So catalase, and if we don't get to this lab safety, it's not a problem because we're not really making anything dangerous. Okay. So catalase is an enzyme that breaks down hydrogen peroxide. So here the substrate would be hydrogen peroxide. Enzyme is catalase. That you should know by now. And it tells you all that here. So they want you to label everything on the next page. Actually be doing, so you should be very aware of it. And at this point, I think everybody has it. But this should be catalase. Catalase doesn't change, so all three of them are catalase. This is H2O2. This is the enzyme substrate complex. Literally, it's in the name, enzyme substrate. These are the products. The enzyme is as it was, and what you're producing is oxygen, and water. Label the chart here. This is the amount of energy necessary for the reaction to work without an enzyme. We call that amount necessary is called the activation energy. So that's also here. And this is the amount of energy necessary with an enzyme. And we call that, again, activation energy. Of course, you start with reactants and you end up with products. You start with reactants and you end up with products. Yeah, I'll be posting as soon as I finish it's interesting to point out that I'm going through the explanations here and going through the answers, and people are not writing it down, and this is due on the day of the test, right? Say what now? I can't hear you, I'm sorry. pH, remember, and I did describe it earlier in the video, but I'll describe it here because you asked. pH is equal to anywhere from 0 to 7, from 7 to 14. So a pH from 0 to 7 is called an acid. If something has a pH of anywhere from 7 to 14, above 7 to 14, we call it a base. If the pH is 7, we call it neutral. You say something to me? Tissue, no. We say it's neutral, so it's not acid or base. So that's how much hydrogen ions are in the solution. So here there's high hydrogen ions, and here it's low hydrogen ions. So if you have a lot of hydrogen ions or too little hydrogen ions, that impacts hydrogen bonding. So it changes the shape. So that's pH. And if you watch the video, I go over pH again earlier as well. So it says, how does blank influence the rate of catalase breakdown? Well, you can fill in any of these. So if you pick one, let's say heat, how does heat impact? So let's just choose, we're going to have to choose one or two. So let's just choose heat as one. And if we choose heat, the potential research question might be, does increasing heat always increase the rate of reaction? So if I increase the heat, will continuously, what will happen if I keep increasing heat? Do I expect it to do this, keep going up forever? Do I expect it to stay the same? Do I expect it to go up and then come down? Like, is something else going to happen? Which one of these is going to happen? Let's find out. Let's do it. Let's change the heat. Let's keep everything else the same. Potential question number 2. Let's choose enzyme concentration as the second one. So does increasing the concentration of catalase continues always increase the rate of reaction? So again, which of these is going to happen? Am I going to have this, or this? I could have all kinds of graphs could happen. Maybe I don't have any of that. Maybe I have something that does, something like it does this. What if it flattens out? Just keeps the same after that. It doesn't increase, but doesn't decrease. So in the first one, it stays the same. In the first one, goes up and keeps going up. And the second one stays the same, no impact. The third one, you see that this one means no impact, right? The third one, it goes up and then comes down. And this last one, it goes up, but then it flattens out. So which one of these is going to happen? Or is there some other? All right. What's the last one you want to do? Which of these do you want to do? We have substrate. You want to do pH. All right. The last one then would be, does increasing the pH increase or decrease the rate of product formation? So now you have to choose which one of these are you going to write for the rest of this. So I started you off. I'm going to finish this with the other classes because how much time do we have left in this class? So we have 5 or 3 minutes. I hear 2 different numbers. 5? So how much time? Okay. And soon it'll be 1 and 0. Okay, so I'm going to keep going until time's up. In the next class, I'll continue. Follow your teacher's procedures for determining the research question you will investigate. Pick one. I don't care which of these you pick for your experimental design plan. We can do all 3 of these or one of these, whichever one you want to do. Just pick one. I'm going to pick heat, and I'm going to walk you through the answer for heat. Number 2. Let me do a different color. A, B, or C. I'm going to pick C. No, I'm sorry. A as my question. Once you have a research question, complete the experimental design form. You will submit the form to me on finals day. Again, you'll have time during the day on finals day to do this, but if you're waiting till the day of finals to do this, you got problems. So you're going to come in on Monday, you're going to take your multiple choice test. So first thing you're going to do, you'll have an hour to do that. Then I'll say, complete your experimental design. Yeah, you write on a separate sheet of paper. I would use pencil and just use the form that you have here. You can rip them out and hand them in. Staple them together, make sure your name's on it, the date. So once your experimental design is approved, gather the additional materials you need to carry out the experiment. So let's do your experimental design. So your research question, hold on a second, so I just pasted this from the previous page. Does increasing heat always increase the rate of reaction? So the null hypothesis would be if increasing heat has no impact. So null hypothesis is the increase heat will have no statistically significant difference. So if the null hypothesis is true, that means increasing heat will have no impact. It won't change how much product I make in the same amount of time. In the case of this example, this setup, that means the bubbles won't form any faster. Again, I'm going to finish this today and post it. You can watch it over the weekend. You can have this part done and just discuss it amongst each other before you submit it for grading at the end of Monday. So the alternative hypothesis would state that something will happen. There will be a statistically significant change. So if we think about what we're doing, we're going to increase the heat, and we think about all the things that we talked about before, the impact on heat on enzymes and the impact of heat on chemical reactions. So there's two opposing forces working here. First, with any chemical reaction, when we look at a chemical reaction, any chemical reaction, when you're adding heat, what you're doing is you're moving the particles faster. And since you're moving them faster, because remember, heat, or as you increase heat, you increase temperature. And temperature is a measurement of average kinetic energy. So the particles are moving around faster, and because they're moving on faster, they're going to find each other. And if blue particle meets up with green particle, then you have a chemical reaction and you form something new. That's a reaction. In this example, we're talking about breaking something up. We're talking about catalase. So you're talking about a sugar, which, by the way, this disaccharide is in fact two. It's glucose, which is a hexose, and it's also galactose, which is also a hexose. So these two are together, and then you could increase heat, and that would increase the amount of energy available. So you could increase it to the point where they would separate on their own and become two new things. Instead of lactose, you would have glucose, and then you would have galactose. So you'd have two new things instead of one, lactose. You'd have two things in the end, all by itself, just by increasing the amount of heat. As we said in class, that is not a good thing for living systems, because you'd increase all the reactions that way. There's no way to increase only one reaction by increasing the heat. Everything would increase. And you don't want that to happen if you want to have control over what's going on. Right? Nobody here wants to eat and poop and sweat and shiver, right? All at this, and run and walk and sit all at the same time. Correct? So as a simplified form of what we're talking about here, you want to control the chemical reaction so you have only one thing happening when you need that thing to happen, and be able to not happen when you don't want it to happen. So it gives you control over the chemical reaction. Breaking up lactose, a mother does not want to break up the lactose. She made the lactose. She ate glucose and galactose, and your mother made lactose in her breast and put together glucose and galactose and made lactose along with the fat and the protein that's in milk so that she could feed you as a baby. Then you drink it, then you want it to break up. You don't want it to be broken up before you get it, and it wouldn't work. So in any case, you want to control the reaction, so you need an enzyme. So temperature would increase the chemical reaction. But also, obviously, the way our bodies work is we don't use temperature. What we use is an enzyme that fits where that has an enzyme that fits lactase where lactose fits into it. And remembering that at the end, I'm just trying to review stuff that you're going to see on the test, that the lactase you had in the beginning, you have it at the end. It's just you took lactose and you split it up. Does that make sense? Is that making sense, yes or no? So the problem is that if it gets too hot, if you remember what we went over yesterday, this enzyme, there's a third thing that you have to consider, because all of it together is the only way you're going to understand what's going on. If you add too much heat, too much, obviously some is good, because with some kinetic energy, the molecules are moving around and they'll find each other. With some kinetic energy and an enzyme, it works much faster because the enzyme brings them together and creates an environment where they can be broken up. But when you add too much heat, I'm sorry, I don't want to interrupt you. All right. So when you add too much heat, the enzyme changes its shape, and this is lactase with too much heat. So here, lactose won't fit. And because lactose won't fit in the active site, remembering, I just want to mention again, the active site is where the substrate fits the enzyme. Before too much heat, it's not a problem. Adding too much heat, now the enzyme, the substrate, does not fit in the active site. In this case, it's lactose. Doesn't fit. So the reaction rate won't work as well. Does that make sense? So how do we phrase that as an answer here? So the way I would phrase it, put all that together by saying, well, good, that lactose won't fit. That's perfect. That's perfect. That's right. The way I would phrase it, though, is I would say increasing the heat, should increase the reaction rate until the heat becomes so great that the enzyme loses its shape. At that point, the reaction rate should decrease. If I wanted to do a graph what I would expect to see in a graph is as time, if this is product and this is temperature. I'm controlling temperature, so this is the independent variable. The dependent variable is how much product I'm producing. What I would think would happen is that in the beginning, if I'm not starting at zero, if I started zero, it would be zero product because it would be frozen, right? So nothing would be happening. By the way, that's why we freeze our food, right? So that it doesn't change, doesn't go bad. You take it out of the freezer, sitting on your countertop, then all kinds of chemical reactions are happening.
So the control variable would be
well, let's talk about the control in a minute, but first let's say that what I would expect to happen if I took these words I just wrote and put them on a graph, I'd expect that as the temperature increases, the reaction rate would increase in some way until I get to some point where then it would start to decrease again. So I expect to see something like this. I'd expect that there would be a temperature range that would be ideal. There would be an ideal temperature range.
Why would it decrease instead of just keep going up?
Why? What happens to the enzyme when it gets too hot? It changes form. So is it going to keep speeding up the reaction? No. So because of that, the enzyme is not at this temperature. So that's a good question. Let's think about it. At this temperature.
So that temperature is the highest it can go without changing form?
That's right. So between here and here is the max range, or is what we call the ideal range. Right? Is between these two temperatures. And the max temperature is obviously going to be right here, right? Does everybody agree? That's the best you're going to get. Then you start decreasing product after a certain point, and that's because the protein's denaturing. The word is denaturing, by the way. So protein, here the protein, we call it an enzyme, is losing its shape. It's becoming less and less a good fit, so it's becoming less and less productive. Over here, it's just too cold. Not enough energy. So right in the middle, the Goldilocks range is right there in the middle. This temperature here, what is this temperature? Well, it all depends. The temperature from here to here. What is this ideal temperature? Well, let's think about it. It could be a lot of different things. If it's in a human, what temperature range do you think it would be the best? Yeah. That's right. Something, I'm just going to give a number. Whatever your average temperature would be. So let's say 97.5 degrees Fahrenheit would be ideal for a human enzyme. Another enzyme, something that lives, if the creature lives in an area that's, let's say 100, let's say its normal body temperature is 100, what temperature do you think range would it be? What would be the max? Yeah, something like that, somewhere around 100, right? By the way, obviously, when I say 97.5, I'm looking at the max right here, right? So for the max for this creature that lives in an area where its body temperature is always at 100 to 105, its max would probably be 100 or 101 or something like that degrees Fahrenheit. If the creature lives if its body temperature is normally 150 degrees, then its temperature, its enzyme would probably be around 150 degrees Fahrenheit. So at what temperature does this enzyme work best? It all depends on the creature and its own body temperature, because you're making your enzyme, right? You wouldn't make an enzyme that doesn't work at your body temperature. That doesn't make sense. So, what is the independent variable in this experiment? Again, if you pick another question, it would be a different independent variable. The independent variable will be the temperature, because it's the one you're changing. So, what temperatures what are the levels of temperature? Well, we're going to use a human catalase. So we probably want to have something at like 30 degrees. And then we might want something at around 60. Okay. 60 degrees, a little warmer. That's still cold, so let me just keep it blue. 60 degrees Fahrenheit. All these degrees Fahrenheit. Then we might want something that's near 90, let's say 97 degrees Fahrenheit. And maybe we have something like crazy, like let's do 120 degrees Fahrenheit. And see where it goes. Now you can pick anything. You can pick 30, 97, 120, 150, right? Obviously, boiling is not something you want to go to. Probably not a good idea. Number of repeated trials. So you probably want to do, just because of how much time we have in class, we probably want to do 2 trials here, 2 trials in each. And then if we have more than one group doing it, then we'd have multiple trials, right? So if each group is doing 2 trials and we have 10 groups, that's 20 trials for each temperature range. So we're pretty good. The dependent, what are we measuring? We're measuring the product. Now, this goes back to trying to explain what we're doing. You're going to have a water bath, a container that has water in it. And then you're going to have, at the bottom, you're going to have this flask that has a stopper on it. Obviously has a stopper because you don't want water to get in. There's a glass tube that sticks into the flask, like this. And that tube goes all the way through, but it's sealed. So notice that the water's not getting in, correct? Then you're going to put inside here, you're going to put hydrogen peroxide, H2O2, hydrogen peroxide, the same stuff you put on your cuts. And you're going to add to that some enzymes. What enzyme are you adding? What enzyme are you adding? Who's been paying attention? What enzyme are you adding? No.
Is it the glucose and the
No. No.
Wait.
You're adding catalase. So in the example we were talking about earlier, here we're dealing with lactase, right? So this was an example we were using. I believe here we're using H2O2, are we not? Are we still talking about lactase? No. Here, this is an example from the previous situation, correct? When we looked at lactose being split into glucose and galactose, right? Yes? But the experiment we're working on here is looking at catalase breaking up H2O2 into 2 things, into oxygen gas and water. That's the experiment you have to deal with on Monday, right? Or Tuesday in your case. Yes? We're not going to use milk. We're not using lactase. We're not using lactose. We're going to use hydrogen peroxide. And we're going to make oxygen gas and water. So you have a rubber tube that connects to the glass tube. It'll go around it like this, and the rubber tube's. Oh, let me use a different color. You have a rubber tube. It's going to go around there. It's going to go into the water, and it's going to come up here. And the rubber tube is going to go into a graduated cylinder. A graduated cylinder is just literally what it is. It's exactly how it sounds. Just a cylinder that has graduations on it. That's marks. Marks at a regular interval. And of course, these will be in milliliters. And this might be, and at the top, the graduated cylinder is going to start with being full of water. In the beginning, this is going to be full of water. This is the setup that you were supposed to have read and be ready to deal with in class on Monday. So you should know what this setup is. That's why I wanted to do this experimental design with that particular experiment. So this is going to produce what? And is the water going to jump up here and go down through this tube and come up here? No, it's going to be a liquid water, so that's not going to happen. But the oxygen gas is going to go into the air, correct? And you're going to have oxygen gas going into the air. And where's it going to go? Into the tube. And it's going to keep going to the tube, and then it's going to do what? Into the graduating cylinder. What's going to come out over here? No, water? If oxygen gas is coming here, what's coming out of the tube over here? Oxygen, what? What is it going to look like? Bubbles. The bubbles are going to start. Are the bubbles going to go down? No, they're going up in the water. So they're going up in the water. And what are they going to do when they hit the top? Well, I guess they could pop, but they won't. As it keeps filling this up, what's going to happen to the water? It's canceled. This is sealed. The opening's down here. It's upside down. This is the opening. It's all filled with water. Huh? A layer of bubbles at a time, and bubbles keep coming. What's going to happen? Down. That's right. What'll happen is the water will keep going down as the oxygen gas fills up this container. The water is going to keep coming down and down and down. What's pushing the oxygen? What's pushing the water out? The oxygen. The oxygen gas. Does that make sense? So as the oxygen gas gets filled in, this water is going to keep getting pushed down further and further. The bubbles will burst at the top and fill up that space. And as more gas fills in, that pressure from that gas is going to keep pushing the water level down. Water's going to keep coming down. The water level's going to keep coming down. And the more gas, this is how much gas you have. You can literally read it right here. If this says 10, if this says 10 milliliters, then, and the water level gets to here, then you have 10 milliliters of gas in that amount of time. Does that make sense? So you can measure how much product, how much oxygen gas you're producing in milliliters over time. Just simply by seeing how much gas fills in this space and pushes it down. How do I know what's pushing this down? It has to be oxygen gas. It can't be water. The H2O2 is over here. The water's over here. Unless the water's steam, which it's not going down. So.
Wouldn't the water go back into that heat tube too? Or would it just go straight out?
Oh, that's a good question. The gas is pushing out this way. Is the water going to come down in this way?
Yeah.
No, if the air's coming and pushing this way, the pressure's going down. This gas is pushing, and it's pushing in this direction. Is the water going to go down this way?
You're saying after overflow?
Well, it won't. Let me just say this: it won't. The gas, the pressure, the air pressure, the gas, the oxygen pressure will push everything in this direction. It'll get up here, can't go anywhere, so it's going to push the water down. But no, the water will not go into the tube because there's more pressure pushing up than is pushing down. There's more gas here than there is up here. Is that clear? Mm-hmm. So there should be no water coming down in this tube. All the bubbles will just keep coming up. All right, there's that. So what is the dependent variable? The product will be measured. Measured in milliliters of oxygen gas which is one of the two products made. Right? We're making two products, oxygen gas and water. We're not going to measure the water because it's hard to measure it because this hydrogen peroxide is the same color as water. You can't see the difference. But we can see the water getting pushed down in the graduated cylinder. Is that making sense to you? If hydrogen peroxide were yellow and it was turning clear, then we could measure the change. But because we have no way of telling is this water or hydrogen peroxide with our eyes, we're going to use our eyes to see how much water is pushed down in the graduated cylinder. All right, so independent variable, dependent variable. What factors are you going to control? You obviously have to keep the time. I think it should be obvious. The time has to be kept the same. Right, so you're going to spend 10 minutes on each trial. Then you see how much gas in 10 minutes did you make. I say 10 because it's always easy to divide by factors of 10, right? Just move a decimal. So that's why I always use 10 or 100 or 1,000. As you know, with your assignments are always 100 or 10 or 1,000 because it's easy to divide and multiply by that. You want to keep the amount of enzyme in this experiment the same. Constant between the different trials. You want to keep the amount of hydrogen peroxide the same. And then the tubing as many other factors as you can. But those are the three big ones that I would think might impact your experiment. And they say 5 to 8, so I might as well add 5. Those three are certainly the three most important. I 5. Another one is the level of water used. And the start level in the graduated cylinder. So if you start off at different levels, somebody might read it wrong, or it could lead to errors. So it's probably best to start at the same level in the cylinder. What are the control groups that you're going to use? Well, what I would use in a control group is control group 1. I would have obviously in my flask, I would have an experiment with everything else being the same. They would have H2O2, but no enzyme. And I would have this exact setup, but then no H2O2. I would just had enzyme and water. No H2O2. And I would increase the temperature to the same temperatures. And so I'd have 2 flasks in there. There'd be 3, right? 3 different trials there for your control groups. So one control group would be no enzyme. Same temperature and everything, just no enzyme. 2, no H2O2. Yeah, I think that would be fair. All right. Procedure modifications, describe modifications you would make the initial protocol of your investigation. I don't think anything else would change other than you're adding 3 different temperatures. Data collection, create the prototype data table. Well, you would have temperature. You'd have temperature, and then you'd want to see how many milliliters were formed for each temperature, right? So you have temperature 1, temperature 2, temperature 3, and temperature 4. And then you have how many milliliters of oxygen gas. So how much in each trial. And then of course you do that various times, and all that for let's say 15 minutes each. And you see how much gas is produced in each one of these different temperatures, keeping the amount of enzyme, the amount of H2O2 the same. Type of graph you would use. What type of graph would you use? Let's see. Let's take a look at it. We have 4 different temperatures with 4 different milliliters, and we'd want to compare them. We're not measuring time, but we could do a bar graph, or we could do a line graph or a scatter plot. And the way you could do either one is if you set temperature and you kept measuring it each time you increase the temperature, But because you have 4 different temperatures and you're comparing them, I think a bar graph would be the best. 1, 2, 3, 4, and you'd have milliliters. You'd have 4 different bars. And then you'd predict your results. I think I'll leave those 4 questions for the next class. You all should have everything you need to have a correct answer for this part of the final. So when we're looking at this, we're seeing predict what your results would look like. What will your results look like if you fail to reject the null hypothesis? Now, this is that double negative stuff that everybody hates about science, and to some extent statistics. When you have the null hypothesis means that, so you have to break this down. The null hypothesis means that there is no relationship. Let me make that a little neater. No relationship between the 2 variables, right? No, or in other words, the opposite of what your alternative hypothesis is. No statistical relationship between the 2 variables. Test it. No relationship between the 2 variables tested. So what would it look like in order to reject it? What would rejecting it look like? If we rejected that there was no relationship between them, that would mean that there was a relationship between them. If you reject the null hypothesis, that means there is a relationship. What could that relationship be? Well, that relationship can be a negative relationship. In other words, as I increase temperature, it decreases reaction rate. If it could mean a positive relationship. It would look something like that. It could mean a relationship that's more complicated, so it goes up and comes down. It could mean a relationship that's the opposite, that starts high and goes down and comes back up. So what does that relationship look like? But all of these show if you had any of this going on, you would have to reject. That's the rejection of the null hypothesis. Because none of these show no statistical impact of temperature. Because remember, our independent variable is temperature. None of these would show that temperature does not impact the product, right? So all these would be products. So what would show us that the temperature doesn't impact products? What would allow us to then reject the null hypothesis? Well, that would mean is as we increase temperature, the product did not change. The amount of product we produced over time wouldn't change. So then it would look something like that. That would be no impact of temperature on the reaction. Okay. So if you failed, now see, this is where it's so annoying. If you fail to reject the relationship, that means that the fact that there's no relationship. So so rejecting would mean that there was a relationship. to fail to reject, it means that there is no relationship. So in diagramming this sentence, you see that they have a lot of thinking to do and just to understand the question. So now when we look at what would the results look like, I would expect to see no change in oxygen produced as temperature changed. All right. So what I'd expect to see is there's no change as temperature change. That's it. What will your results look like if you reject your null hypothesis? So if I reject my null hypothesis, what I would expect to see is I would expect that as temperature that at higher temperatures I would produce more gas. And or rather until the temperature where catalase denatures is reached. Then I expect little to no gas produced. So what I expect to see when I look at this is I expect to see an increase in, I don't expect if I'm accepting the alternative hypothesis, right? So the alternative hypothesis is being supported. I'm supporting the alternative hypothesis. Then I would expect the data to show, when I graph the data, if I graph it in this form, I'd expect that the data would show that as I increase temperature, the reaction rate would increase. Then I'd reach near its, at some point the temperature gets so warm it starts to denature the protein, and the enzyme's not going to work as well, and eventually it stops working altogether. I expect to see some kind of temperature range where we're at the peak, and there'd be some max point, and it's this temperature range. I'd also expect it to be at the body temperature of the organism that I'm measuring. So I would expect that this whole temperature range, which is the maximum temperature range, the maximum product I'm going to get, the temperature between, the here to be the temperature, this would be the ideal temperature range for this enzyme. Before that, this is just too cold, which means too little kinetic energy for the reaction to really take off. And here, the protein's denatured, and that means the substrate does not fit the active site anymore. All right. So that's the whole point of how I got to this answer here. All the stuff we've been talking about is that denaturing, I would expect it to go up and come down. So if it is supported, I would write it out. The way I would write it out in a paragraph form would be, I expect the oxygen gas, because remember I'm measuring oxygen gas volume to increase Well, this is actually, this question is actually easier to answer, because all I would, honestly at this point, what I would say is same as 2. Because when you reject your null hypothesis, right? Reject your null hypothesis, you are accepting your alternative. So this answer for number 2 is the same as answer number 3, because rejecting the null hypothesis means accepting the alternative. There's only 2. Either you think there is a relationship or you think there's not. If you're rejecting the fact that there isn't, then there is. So these 2 would be the same. Just 2 ways of saying the same thing. I'm rejecting the fact that there is no relationship between the 2 factors, and I'm accepting then that there is evidence that they are related, that one is impacting the other. What will your results look like if your alternative hypothesis is not supported? Well, then if my alternative hypothesis is not supported, then it would be the same as number 1. Now I'm back to keeping it simple and saying same as number 1. Because when I say alternative hypothesis is not supported, that means I'm accepting the null hypothesis. Accepting the null hypothesis is the same as not rejecting or failing to reject the null hypothesis. It's just word play is what this is. So you have to break this down and think about it logically. It's really confusing, but they do love to play this game. So please be careful on how you're answering and reading the question and really thinking about it, breaking it down. If something says not, some key words is not, look for those nots. And it says it looks like if your alternative hypothesis is not, right? And then the word supported, that's the same as not rejecting, fail to reject your null hypothesis. It's really horrible for them to play these mind games with you, but that's what they do. All right. So be careful. It's horrible, but yet again, it's useful. It makes you think, and it makes you really consider the meaning of alternative hypothesis and null hypothesis. So it's a good exercise. I would just hope they wouldn't do that to you on the final. All right. These are just reporting tools that you're going to report after you've decided. This is all for after the final. Next, when we get back, when you are doing a lab, you're gonna, and in the future you'll see some of these. You're gonna write out what your question was, the balanced chemical reaction. If you decide to do, we're doing H2O2 turning into water and oxygen, but just to keep it simple. But you could do all kinds of experiments with all kinds of different chemical reactions. And then here, obviously, we are talking about H2O2 and oxygen being produced on the y-axis. You put oxygen being produced on the x-axis. You have time in seconds. And here you're going to just measure how much time it takes to make oxygen, the oxygen being produced every so many seconds. So what I would do is every minute I would take a measurement. So if this is if we're talking about 1 milliliter, 2 milliliters, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, whatever it is. And how you scale this is going to depend on the data that you have. I'm just making up the data and say it's somewhere between 1 and 20. Then I'm okay with that. And then I would take every minute, I'd make a measurement. So if I did every minute, and this is in seconds, so when the independent variable here is the time that is going to be time, that's the thing that's not dependent on the other, right? In seconds. So then, taking a measurement every second is ridiculous, but if you do it every 30 seconds, so 30 a minute, right? A minute and a half, 2 minutes. So, you put the number of seconds here. So 30, 60, 90, etc. And so if you start the reaction, and then after 30 seconds, you might get, maybe half a milliliter of gas being produced. And then after a minute, maybe you have, a whole milliliter made, and then some. And then after, you might then, see something like this happening, and then like this, and maybe something happens and there's that. And I'm just plotting points here that might come close. And again, I'm changing the temperature, so the temperature is going to be the same. So maybe the blue here, the blue dot for my key is going to be at 60 degrees Celsius, and then the red dot might be at 97.5 degrees Celsius. And then, you know what, I shouldn't use, not use red. Let me use maybe a green might be 97 degrees Celsius. And then for red, I might use 150. And I'm saying Celsius because I'm so used to saying Celsius, but let's just use Fahrenheit. I'm using Fahrenheit because you guys are used to hearing the degrees in Fahrenheit. And then if I say 150 degrees Fahrenheit, then I kind of expect that I could have a 60, at 60 degrees, I might get something like, over time. And something like this. If I'm changing temperature, right? And then I draw my best fit line. And my best fit line is something that looks like, I don't know. Something looks like this. It's not necessarily through all the points, it's just as close to all the points as I can get. And it would be theoretically a straight line, which is not looking too straight right now. All right, something like that. Notice that some of the points are off. That's fine. That's going to happen. The best fit line does, and it's not straight, but that's because I can't draw a straight line with my free hand. So, but it's close. Now, if for my 97, I shouldn't have used a green arrow line. Actually, now that I see that, I should use some other color. I'm just use a blue line. So if I draw a straight line, as straight as I can, anyways, something like that. I don't know. I wish I had. Well, you know what? I have a ruler. Let me. That's about as straight a line as I'm going to be able to get. Then if I made 97 as 97 degrees, I'd probably expect it to start about the same place. And then as I measured, I kind of expect it to be even faster at doing that. And if I did the best fit line again, I can see that the slope of this line is better or steeper than this one. So at this point, what I see from the evidence that I've gathered is that this the 60 degrees Fahrenheit, that 97 degrees Fahrenheit is better for the enzymes working, the reaction, than at 60 degrees. Then if I did 150 degrees, what I'd expect, honestly, is pretty much zero across the board. I'd expect to see no gas produced at this point. Or, you know what, that's not true for hydrogen peroxide. I'd expect something to be made, but I don't expect it to be very, very quickly at all. I'd have to look up what the boiling point of hydrogen peroxide is, but unless I hit the boiling point of hydrogen peroxide at 150 degrees Fahrenheit, I'd expect something like that, maybe even less steep. Something like this, maybe even. And this would be the worst. Might even get a flat line, depending on what the boiling point of hydrogen peroxide is. Remember, boiling means it's turning into a gas, but there we're not measuring oxygen gas, we're measuring H2O2 gas, so it gets complicated. Because here, when we're measuring the gas produced, what we're really trying to find out is how much of that product, the reactant, became the product. How quickly that happened. I'm looking at how fast does that happen. And I'm looking at rate. So when I'm looking at the reaction rate, I need to know how many milliliters of oxygen per second. Now, my time is in seconds, so that makes my life easy. My time's in seconds. So how am I going to find the rate for each of these? I have to do the slope. Now, slope, some people think of y equals mx plus b, but that can get you the slope if you know x, y, and the y-intercept. So x, y, and the y-intercept, then you could calculate slope. So you just need 1 point, an x and y. The y-intercept, you can calculate slope. Or I think the generally more accepted way is to take a look at change in y and divide it by change in x. And when you look at x is time. So since x is time, then you're looking at the bottom being change in time, which is in seconds. And y is how many milliliters of oxygen which is your product. You're also producing water, but we're not measuring that. And you got to remember when you see that this symbol delta that equals final minus initial, so change in y, now we're going to have to pick 2 points. It doesn't matter which point you pick. The only thing that matters is that you keep the points consistent. You keep their order consistent. So 1 and 2. Now, I'm recording something. So when you're recording, let's see, interruptions. They just mess me up every time. So if you're looking at the slope, and you're trying to find the slope of the blue line, for instance, then you would look at first the y. So in this case, it would be 1 is going to be 2, which equals, so in this case, you'd want to have change means x or y2 minus y1 divided by, that's what change means, and x2 minus x1. So that would mean, for this case, it would mean 15 minus 2, and this x2 minus x1 would be, just guessing here, 370 seconds minus 170 seconds. I'm just making up these numbers. Obviously, there's a class coming in. And so when you divide that, what you get is 13 divided by 200 and minus 200. So that means that you end up with 0.065 milliliters. Now, keeping the units the same, this is in milliliters of O2. That's what you're measuring there. Here's in seconds. So this is milliliters of O2 of oxygen gas, and this is seconds. So this is milliliters of oxygen gas per second. So notice that what you have here is the change in volume of gas produced in a given amount of time, right? And that is a rate. That's a rate, and that's all rates are slopes. So it's a rate of change. Rate of change is how much does something change over a given amount of time. It's generally, a rate is generally the slope of a line. All right, now that these rude children have come in, these rude children using bad words on something that I'm not going to edit, and I'll make sure to send to their parent, particularly with an emphasis on the time stamp where the bad word was used, so that they can explain to their child why it's inappropriate to use bad language, and maybe to the entire class, so that all their parents could talk to them about how they should come into a classroom. But that completes this discussion, and that gives you all the answers to everything that's due, except for the practice questions that you need to complete to get ready for the final.